So, welcome back to our virtual classroom. Today I am sitting down, trying something a little different. It's weird, but yeah, this is what I'm doing today. So today I will be talking a little bit about DNA, specifically the structure of DNA, both in its minutia and in its chromosome form, as well as looking at the structure of genes within DNA. So it should be a pretty quick talk, focusing on shape and function. Good? Let's get to it. So, first things first, the structure of DNA. As you should know at this point in time, DNA is a nucleic acid, hence the Na at the end of DNA. And nucleic acids are built from the monomer nucleotides. Right? Our DNA nucleotides are guanine, cytosine, thymine, and adenine. Right. Forgot that last one for a second. And so these nucleotides are made of three main components. There is a phosphate group, there is a sugar group, and then there is the nitrogenous base. So for all four nucleotides, the phosphate group and the sugar group are identical. Right? For DNA, that is deoxyribose as the sugar, but that nitrogenous base is varied. Right? So there's four options. One of them is adenine, one of them is cytosine, one of them is guanine, and one of them is thymine. Right? And that is what we define the entire nucleotide by, is that varied nitrogenous base. Right? And now the nitrogenous bases carbon rings. Right? So we have the purines, which have 2 carbon rings, and we have the pyrimidines, which have 1 carbon ring each. Right? And a pyrimidine always pairs with a purine. So it's always 1 plus 2. So no matter where we are in the DNA double helix, we are 3 carbon rings wide. Okay? A, adenine pairs with T, thymine, and G, guanine pairs with C, cytosine. Okay? This pairing across the 2 strands is achieved through hydrogen bonds, so it's a rather weak pairing. Okay? And between G and C, there are 3 hydrogen bonds, and between A and T, there are 2 hydrogen bonds, meaning that the A and T are slightly easier to break apart, which has functional significance when we get to opening up the DNA strand. Right? The 2 hydrogen bonds are easier to break. When you break these hydrogen bonds, right, they are going to go back together. Right? There's millions of these nitrogenous bases paired up along this strand of DNA. And so if you open up a little segment, right, on each side they're still held together. And so those bonds will just reform just as they were. Easy peasy lemon squeezy. So you can open up the DNA and it'll close back up. You can open up the DNA and it'll close back up. No harm, no foul. This is the benefit of utilizing hydrogen bonds to create this binding. Right? Easy to break, easy to reform. Not much energy required. Okay? Yeah. So I will talk about the double helix shape in the next section. So I'm sure you all can imagine the general shape of a DNA helix, right? It goes, and it is 3-dimensional, so it twists in and out as it goes, right? There are actually 3 different variations of the DNA double helix. There's the A variation, the B variation, and the Z variation. The one that we were always referring to is the B variation. It's the predominant DNA shape in living organisms. They all have this double helix shape. It's just a matter of how tight the helix is. Like the A one's really sloppy and kind of messy. The B one's the one that we usually think of, pretty clean. And the Z one's more elongated. Right? The only one we really care about is the B one. So don't even worry about the A and the Z one. The double helix, each twist is about 10 base pairs long. Right? And the twists are slightly asymmetrical. Right? And so it's tighter on one side than it is on the other. And the little spinny space is called a groove. There are two grooves in this helix. Okay? There's the major groove and the minor groove. Okay? And so because it is asymmetrical, right, on the twist, if you think of it like a road, right, you have the inside twist and the outside twist. And so you have taken the inside curve versus the outside curve. The inside curve is really tightly packed, right? It's blocked mostly by the carbon phosphate backbone there, the sugar phosphate backbone. But the major groove, it's a little bit more wide open. You have better access to the base pairs. So one side of this DNA is readily accessible and the other side is not. It's too tight. Things can't get in there. Okay? And so this is important when you start talking about the coding strand versus the other strand for some unbeknownst reason. I can't think of the name right now, but that's okay. We'll get to it at some point. I got the coding strand. That's the one we're actually using. Template strand is the other one, right? Template strand. Okay, but we got the coding strand. That's where we actually need to get to for the genes. And so that is in the major groove side of the helix, and it's readily accessible. Okay, so yeah, I think that's it. All I want to say about the double helix. All right, major groove, 10 base pairs long per twist. Good. So, our DNA is organized into long specific strands called chromosomes. As eukaryotic organisms, we have linear chromosomes and we have multiple different ones. As humans, we have 23 different chromosomes. Right? And so the chromosome structure is this one long single double helix. Right? It goes from end to end. And the ends are called telomeres. In the middle, well, it's not really in the middle. Often in the model, we represent it as being in the middle. But somewhere along the length of the chromosome, there is this tight bundle called the centromere. Right? And the centromere is the location where during mitosis or meiosis, the centrosome reaches out its microtubules and grabs onto. It is also the location that anchors the sister chromatids after DNA replication is finished. Right? So this centromere is a point where the chromatids can stick off, right? Or where the centrosome can attach to for cell division. Right? But it's just a location within the chromosome. Right? Along the entire length of this chromosome, there are histones, which are a protein that is very important for the formation of the specific shape of a chromosome. Right? And so they're proteins that are effectively like ball shapes. You got 4 of them combined to make one histone. So if you imagine 2 balls, 2 balls, 2 balls, 2 balls, right? Stuck together. Yeah. The DNA, the chromatin of the DNA, wraps around the histone. And so it goes from around, it goes around the other ball, and then it comes out the other side. Okay. So this protein is kind of like a fishing reel, is how it works. And the DNA wraps around it. Okay. You can twist it to make it tighter, and so you can tighten the DNA around this histone. You can twist it the other way, it'll make it loose. So you can loosen it and you can tighten it. And we'll talk about those mechanisms when we talk about gene regulation, because it does play a role in what genes are able to be expressed and what genes are inhibited at any given point in time. So along the chromosome, we have many of these histones. Okay, and they give the bendy shapes of the chromosome a very specific format. Right? And so these chromosomes are the same for all of us humans. Chromosome 2 always has the same gene set for all of us. Within the nuclear envelope, right, we have the same anchoring points for these chromosomes, so they're kind of stuck to certain parts of the nuclear membrane. Right? So my gene 2 might be stuck to the northwest wall of the nuclear envelope, right? And then your gene 2 would be stuck in the same place. And so you have this general location for each of the chromosomes that's determined by these anchoring spots that stick the chromosome to the nuclear envelope, and then they fold out into the nucleus, where they are wrapped around histones, which give them the same general shape. Right? And so our chromosomes all have their own specific kind of general area where they're always found, and a area, or rather a shape, that's typical for each one. Right? And so the outline, the format, the blueprint for how our chromosomes are organized in our nucleus is effectively the same for all of us. Right? So that's important for maintaining a uniform expression of genes as well. Yep. Chromosomes, I think that's it. So our entire collection of DNA is our genome. Right? The genome is the entirety of the DNA that an organism has. And parts of that genome code for protein creation or regulatory mechanisms that regulate protein creation, and those are called genes. And a great deal of the genome doesn't code for anything, and those are called non-coding regions. And so all of our DNA combined is our genome. The entirety of our genome is about 3 billion base pairs long, and something like 98% of that 3 billion base pair set is non-coding. So most of our DNA isn't actually coding for proteins. That doesn't mean it's not doing anything. The non-coding regions actually play a very significant role in gene expression. The understanding of that role is still really naive in the field of genetics, right? We really don't understand a lot of how that works. One way that we do understand about how that works, especially in eukaryotic organisms, is just the spacing between genes and some of their regulatory regions, which we will look at first in the gene structure section I'm about to talk to in a couple seconds or a couple minutes. And then more specifically, when we get into the regulatory mechanisms, we will look at how these non-coding regions are important for the spacing of our genes. Because as you know, shape determines function, and spacing is part of shape. Okay? But there are definitely other mechanisms at hand that we do not yet understand. We do know that if you break a non-coding region, it can very easily ruin your gene expression, even though it seems like it shouldn't because there's no genes there. Right? We also know that non-coding regions have been conserved through millions of years of evolution. And if the specific order of their nucleotides wasn't significant, then they could mutate and that conservation wouldn't happen. Right? The only reason why that would be preserved through evolution is if the order is significant. So there is something important about these non-coding regions for our genetic expression, but we don't really know exactly what that is yet. Okay, there are about 20,000 genes in our genome that code for proteins, and about 22,000 other genes that code for regulatory mechanisms, different types of RNA that regulate whether or not other genes get expressed. So there's that. I got some notes here. Usually I don't have notes, but numbers aren't my best thing. So I got some notes. Any given gene that codes for a protein, the length of that gene will be determined by how complex the protein is that it's coding for. Right? That's logical. That makes sense. One of our shortest genes is 780 base pairs long. One of our longest genes is 220,000 base pairs long. Right? And so these genes can vary in length by great amounts. The average gene is about 67,000 base pairs long. Okay? Just to give you some numbers for y'all who like details. Obviously, I don't remember details very well. That's why I got notes. Yeah, so genome, we are diploid, right? Meaning we have 2 copies of every chromosome that we have. One came from my mom, one came from my dad. When we make gametes, those gametes are haploid. We achieve that through the process of meiosis, right? But we have 23 different chromosomes. Okay? Chromosomes 1 through 22 are autosomal, meaning that both copies are identical as far as structure. Obviously, there's slight variations for genes and stuff, which we'll get to in the inheritance section. Right? But the 23rd chromosome, the sex chromosome, is allosomal, meaning that the two copies can be different, right? Me as a male, I have an X and I have a Y. They're different shapies, right? You know, if you're a female, you got an X and an X. You get the same thing, but it could be different, so it's technically allosomal. I think that's it for chromosomes. Good. Moving on to gene structure. All right, so I thought I was going to talk about gene structure this video, but I'm already hitting close to a 15-minute mark, and I am trying to keep these shorter for y'all, so I'm going to cut it there. I talked about the structure of DNA a little bit, talked about chromosomes a little bit, and talked about the general concept of a genome in this video. Next video, I will focus on the structure of genes, both eukaryotic and prokaryotic genes, and take a look at how the DNA is organized in a way that allows it to be regulated and code for proteins. And yeah, that's it. So, have yourselves a fantastic day. See you soon. Bye.